everybody. We're now on part 2 of our biomolecules lecture. In part 1, we talked about how biomolecules are made and broken down. We took a look at carbohydrates and lipids, and that pretty much was the end of part 1. In part 2, we're going to delve into both proteins and nucleic acids, so let's get started. The first biomolecule we'll mention today is going to be the proteins, and as you can see, they account for more than 50% of the dry mass of most cells. So you can see how hugely important a protein is to the cellular environment. Now, of course, different types of proteins will exist and they will have different roles, as you can see down the leftmost column on this table. We can see enzymatic proteins. We mentioned an enzyme earlier in part 1. Structural proteins, transport, hormones, receptors, contractile and motor proteins, and then finally defensive proteins. All of these proteins are going to work in a perfect harmony within living things, and they're going to keep organisms safe, or they're going to get things where they need to go, and they're also going to build the cells that make up these organisms. Now, as you can see in the second column, the function of each one of those is described. Now, we'll be delving into a lot of these proteins in later chapters and later topics. So we're just going to basically talk today about how they're built and how they break down. So, overall, we can say then they're functionally and structurally diverse. All right, let's get into some protein basics and the terms that go with it. First, proteins consist of one or more polypeptides. Now, I want you to think of the term polypeptide kind of how you thought of polysaccharides when we were talking about carbohydrates. So, with a polysaccharide, we had a single simple sugar monomer, and it was connected to a whole bunch of other ones. And once a whole bunch of others were connected, we call it a polysaccharide. Well, the same thing is going to go with proteins once we find the monomer of those. And the monomer of a protein is going to be an amino acid. And as you can read in the next sentence, they're built from the same set of 20 amino acid monomers. So there's 20 amino acids that are going to make up all the proteins in your body. Here is what they look like. Now, what is this thing down there? Well, let's talk about it for a second, because we're going to see the same repeating structure over and over in all 20 of those amino acids. So they're organic molecules. As you can see, they have carbon in them. That makes them organic. And they have carboxyl and they have amino functional groups. So if you take a look at the structure down here, the carboxyl group is a carbon double bonded to an O, single bonded to an O with an H on the end. The amino group is a nitrogen with 2 hydrogens connected to it. And we saw that back when we mentioned the functional groups in an earlier lecture. So we can expect to see that in every single amino acid. Now, are they all identical? No. And here's where they differ. This right here, this R group, this is a side chain. And that side chain is going to be different in all 20 of those amino acids. So that's what's going to make your amino acids differ. But other than that R group, all of this is exactly the same in every single amino acid. Let's take a look at these 20 amino acids based on the different properties that they have. Now, I want you to take a look at the basic structure, the part that's not highlighted in white, for each one of these amino acids. Now, notice it's identical in every single one of these molecules. The thing that's different, though, remember that R group. We said R was the side chain. Now, why is it on top? Remember, these organic molecules, they're floating in 3 dimensions. So in the previous slide, they were on the bottom, but all it would take is them floating upwards and then they'd be on the top. So don't let that confuse you. Just understand that R group is a side chain coming off every amino acid monomer, and it's what makes one amino acid different from the rest. Now, if we take a look at the white portion, each one of these, the R group, what do we see that's common between all of these? Well, one of the things that I'm noticing is I see a lot of C's and a lot of H's. And when we have a lot of C's and H's, we call that hydrocarbon. And hydrocarbon's bond character was that it was non-polar. It doesn't like water very much. It doesn't like to mix with water. So it's going to get away from water when it has the chance. Now, the other thing I want to bring to your attention as we look at this slide here is that each amino acid has a name, which is right here: glycine, alanine, valine, leucine, isoleucine, and so forth and so on. But we also refer to them, when we don't have a lot of room, by 3 letters. We have 3-letter combinations that will represent represent each of the 20. So just keep that in mind. That will come into play a little bit later when we get into transcription and translation in another lecture. In this second group, take a look at the R group once again. What do you notice? What do you see a lot of in this group? Well, as we look through it, notice I see a lot of O's and H's. Here's an S and an H. There's another O and an H. Here's an O. Here's an NH. And here's an NH. Well, those are more of polar type connections between atoms. So when we see lots of O's and lots of H's, and we have some highly electronegative things like sulfur and nitrogen and oxygen, that is telling us that it's probably going to mix quite a bit better with water. So things that have a lot of O's and H's kind of are like water, because water is H2O. So we say that like dissolves like in chemistry, and if something is very similar to water by the functional groups it has, it's likely that it's going to mix with water. And again, you'll notice each one of these 6 have names, and they have the 3-letter combinations that represent them as well. With this last group, we can point out a couple of interesting characteristics. For example, if you take a look at their R groups, notice that up here you're going to find some charges that are apparent as you look at them. We have a couple of negatives over there, we have a positive over here, positive there. So these are electrically charged. And if you remember from some past science classes, we know that positives and negatives attract each other. So maybe once inside of a protein, these will interact in some way where the two will be drawn towards each other. We could also say that if they have like charges, like two positives coming together, they're not going to stick together like a magnet. If they're forced together, they're going to repel, push one another apart, and not be very closely situated to one another. These are also pH related. We could take a look at how they behave in water and what they do when placed in an aqueous solution. For example, these two on the left, these are acidic, which means that they're going to give up hydrogen atoms in solution. And the ones on the right are basic, which means that they're going to accept hydrogen ions or accept electrons. So in that way, they're pH related. So how do you build an amino acid polymer? Well, back when we were dealing with carbohydrates, we said it was going to be through a glycosidic linkage. When we were talking about glycerols attached to fatty acids, we said that would happen through an ester linkage. And when we link two monomers, two amino acids together, those are going to be called peptide bonds. So the question is, what type of action will connect two amino acids together in a peptide bond? You've seen it before, not once, but twice. It's going to be the same action you saw before. So what was the name when we connected a hydroxyl group from one monomer, a hydrogen atom from another monomer, and water came out? Well, if you're thinking dehydration and synthesis, you are correct. That's how we're going to build these long chain polymers of amino acids. So right here we have our first amino acid. Here's a second amino acid. And here's a third one that floats into the picture, and it heads in towards the peptide 1, 2. Right angle, right speed, and it is able to connect those three atoms together to release a water molecule. Now what we have there is a peptide bond which links the carbon with the nitrogen of the next amino acid that bumped into it. So a polypeptide, therefore, is going to be a polymer of amino acid monomers. Now, these can be short. In this case, we have 3, or they can be long. These could be thousands of amino acids long. Okay, so once you put a few hundred to a few thousand of those amino acids together, you can have a functional protein. So if we were to define it, we could say that it consists of one or more polypeptides. It's going to be twisted, folded, and coiled into a unique shape. And by way of a ribbon model right here, which we sometimes use in chemistry, you can see the alpha helical portions here that look like the old phone cords. And we can see some flat portions in here as well, which we'll talk about in just a second. Over here, we have a space-filling model where we have kind of a spherical shape for every atom that you're going to find in this protein. And by the time you're done, it's going to look like a big blob, basically. And while I mentioned big blobs, in most textbooks, they're going to represent proteins in many different chapters by way in the diagrams of a big purple blob. So 9 times out of 10, if you see something big and purple from a cellular structure, it's going to be representing a protein. Now, this one here has a name. It's called lysozyme. And if you want to take a look and see what it does here, it's a bacteria buster. It finds certain bacteria and it's going to break up their cell walls. So one of the types of bacteria we have is called the gram-positive bacteria. It has this huge cell wall made of peptidoglycan, special type of polymer that enhances the shape and the strength of a bacterium. And what this is going to do, it's going to get in there and break the cross links that hold that cell wall together. As you can see, where could you find it? Many secretions that we have. Tears, mucus, saliva, breast milk. Again, when an organism has a whole bunch of avenues that lead to the interior, there's typically going to be a defense mechanism that's going to prevent them from getting in and doing harm to the organism. So why do proteins take on the shapes that we see as we look at them? Well, first of all, let's talk about the fact that amino acids are going to code for that shape. There's going to be a certain sequence of amino acids, and it's going to be folded up just right to then be a certain shape overall. Now, that shape will determine its function. So it's not as if these proteins are just big blobs that, are any old shape at any given time. Instead, they're very, very specific for what they do. For example, if you look down below here, you're going to see a protein from a flu virus on the right. So that's this yellow and this green thing here. It doesn't sound very good in terms of health for the organism. But over here, we have an antibody protein. Now, it has its own set of bends and curves, and it has this structure down the middle. But what I want you to look at is look how similar these two sides are. It's almost as if this one here fits into that one. And that's not by chance, because antibodies' jobs is to mark foreign invaders for destruction or possibly to decrease the harmful effects they have on the organism that carries it. So every protein is shaped the way it is for a particular reason. A very important role that proteins do have is the fact that they can act as what we called a catalyst earlier to speed up a chemical reaction. Alright, so we have this sucrose up here. We know this better as table sugar. And on its own, it will break down the middle in time in an aqueous solution. However, your body can make that go much, much faster. We have an enzyme called sucrase, and that's what this big purple blob is representing here. Now, this purple blob is going to kind of allow the sucrose to nestle into what we call an active site, and then it might do a little conformational squeeze or change, and maybe it'll hike up that bond in the center upwards a little bit to allow water to get and land on that bond and break it. So when water is added to a particular substrate to break it down, or we could say when water is added to a polymer to break it into little pieces, do we remember what that was called? Hopefully you remember it was the reverse of dehydration synthesis, and this was called hydrolysis. So again, this codes for water breaking. Now, notice the bond is not there anymore. Once the bond is broken, the enzyme will then release the two components, the two monomers that made up sucrose, which are glucose and fructose. They float away for use somewhere else. Possibly glucose is going to be broken down by the body. But look at your enzyme now. It's back to the way it was with empty active sites. And now if there's still sucrose floating around in the bloodstream, now it can land on this and it can be reused and rebroken once again. So enzymes are going to cause that sucrose molecule, this original one up here, to break much faster than if the enzyme was not present. Okay, we're going to analyze these proteins to 4 different levels. So the first level, and we'll be following up this slide here with explanations on each one. Otherwise, it just looks like a lot of written information, and that's kind of annoying. But let's get to it. Primary structure is its unique sequence of amino acids. Secondary structure is going to consist of those coils and the folds we were talking about in a couple of earlier pictures. Tertiary structure is going to be determined by interactions amongst the side chains. Those R groups are going to basically chemically react with each other, maybe pull in tight, maybe repel each other. So it's going to be different as that protein's folding. And the last level of protein structure is going to be quaternary structure, and that is going to exist when polypeptides of tertiary structures come together, 2, 3, 4, or more of them into a big working protein. So let's go ahead and we'll hit these one at a time. All right, so that first level is going to be called primary structure. And as you can see, it's the unique order, the unique sequence of these amino acids. Remember, these 3 letters represent amino acids, but there's a chemical structure that is represented by those 3 letters. Here's the amino end, and here is going to be the carboxyl end of the 25th. So this is 25 amino acids, so 25 monomers, which makes it a polymer. And of course, it's going to be, if you want to officially call it something, you call it a polypeptide at this point, because for these to link together, it's going to take those peptide bonds in order for that to happen. So why do you think, or what do you think the sequence of these amino acids would be based on? If you had a previous bio class, maybe 9th grade, maybe something later, do you remember why these amino acids are in this specific order? Well, we'll get to it in a little bit later, but hopefully we've stimulated some thought there. Try to remember, why would glycine be first? Why would proline be second? Is it written somewhere that it should be that way? More on that to come later. Representing secondary structure, we're going to have 2 major forms. The first form is going to be an alpha helix, which we can see in the background of this picture here in blue. And we said, you know, again, it was that telephone cord shape. And the second structure is going to be what we call the beta pleated sheet. And that's represented by these 2 up here. And you can see there's some nice gradual bends up and down when we talk about the beta pleated sheet. These are peptides of the different amino acid monomers going from left to right. Well, one thing I do want you to notice as well is the amino group of one row connects with the carboxyl group of the next row down through hydrogen bonds. And that's what these 3 dots are right there. So the electronegative oxygen connects with the hydrogen, and that forms a link. So that's going to give it some collective strength as this thing gets bigger. Last thing we can say is that both of these can be found together as in the same polypeptide shape. So it's not as if one polypeptide can only be alpha helix and one can be beta pleated sheet. Often these polypeptides will have both involved with it. The third level of protein structure is going to be called tertiary structure, and that's because we have a lot of R group interaction. Now, what type of interactions are? Well, it depends on, how comfortable you are with the chemistry, but we just mentioned what the hydrogen bonds were. You should be fairly comfortable with those. Ionic bonds we talked about during the chemistry of biology lecture. So when you have things that are positively charged and things that are negatively charged that form ions, they tend to stick together if they're oppositely charged. We also see something called hydrophobic interactions, and that's when we were talking about all of those nonpolar regions mingling with each other. If you're nonpolar and another part of a molecule is nonpolar, you don't have any problem mixing it up with them. However, if you come in contact with water or a polar amino acid, you're probably going to push away and stay away from that polar one. And then lastly, there's some other interactions called van der Waals. So these can be just from moment to moment bumpings of certain corners of molecules, R groups, with each other, and there's a momentary attraction and then a release again. But we can also see some pretty strong ones as well. They're going to hold certain coils and certain parts of the molecule that will kind of fix them in position. A very strong one is called a disulfide bridge, and you can see that here between a couple of cysteine molecules, where 2 sulfurs will bond to each other and kind of lock that portion of the polypeptide in place. Let's take a little bit of a closer look at the interactions between certain amino acids in a polypeptide chain. So what we're looking at here, this is basic. The purple represents the amino acid chain, and what I'll do is I'll break this up a little bit here into various amino acids. So we're going to imagine that this whole thing here, this big purple thing, this is going to be a polypeptide made of different amino acids. Everything sticking off it's going to be an R group. So let's go ahead and talk about each of these interactions, see how they work. So here is the hydrogen bond where we have the oxygen from one side. This is from the R group of this amino acid way down here, coming up and interacting with the hydrogen from this group here. Next, an ionic bond. So this amino acid here has an oxygen with a negative charge. This amino acid has an R group that has this NH3 with a positive charge. And those two are going to attract each other through an ionic bond. The disulfide bridge we mentioned in the last slide, a very strong connection that's going to hold this loop in place. Also, hydrophobic interactions and van der Waals interactions. So here, lots of carbon, lots of hydrogen interacting with each other. Has no problem mingling in that center region because everything's made of the same thing. So this would be a nonpolar region, a water-fearing region for this particular bend of that molecule. So we might find this one here on the inside versus the outside where it might have to touch water. Okay, real quick on this slide, let's show how we go from tertiary structure to the next level up. So tertiary structure is going to be made of alpha helixes from secondary structure, beta pleated sheets from secondary structure, all wrapped up and folded up into a configuration that follows the chemistry of those R groups. That's what tertiary structure was all about. But that's not a working protein yet, so not quite ready. For this protein to work properly, it's going to take some more units, like this one here in the back. There might even be a fourth one there, but this one here is that one. So maybe it takes 4 of those tertiary structures together to make this a successful working protein. So when you have multiple units of tertiary structure coming together, that's when we reach the fourth level of protein structure called quaternary structure. So 2 or more polypeptide chains to form one functional unit. And there's some really famous ones that we talk about often in biology. The first one is called collagen, one of your most common proteins that you have in your body. Structurally, it's very important. As you can see down in the bottom, your tendons, your ligaments, cartilage, your skin, your bones are absolutely full of collagen. Second, we have this one over here on the right. This is one we call hemoglobin. And hemoglobin is made of 4 chains. Here you can see a couple of what we call beta chains, and here we have a couple of alpha chains. So 1, 2, 3, 4 chains. And each one of these has a little spot in the center, little chemical piece that's going to allow this particular protein to do its job, which we're going to talk about a little bit more in the slides to come. We're going to talk about hemoglobin some more as we describe sickle cell disease. Sickle cell is a protein-based disorder. So what we're going to see here is that there's going to be a slight change in the primary structure. What's primary structure again? It was the unique sequence of amino acids that build a polypeptide. And what that's going to do, by changing very little in that sequence, it's going to change the entire structure and the entire function of something that should be working properly in the body that carries it. So this is an inherited blood disorder, and what it's going to result from is a single amino acid substitution. So we saw a diagram that had 25 amino acids. What if one of them is the wrong amino acid? Is that enough to cause something bad to happen? And the answer is yes. One single amino acid causes a person with normal red blood cells to then go from this shape over here, right there is a normal one, to this shape right here, which is shaped like a sickle. And that's where this gets its name. Sickle cell disease means that a lot of the red blood cells are going to wind up looking like this, and they're not going to function properly. Now, what's the problem if a cell is shaped like a sickle? Well, if you look at these vessel diagrams here, notice what's happening at each place where it seems to branch into 2 different vessels. There seems to be some jam-ups, and jam-ups in blood vessels are not good because whatever's downstream from a jam-up is not going to receive the nutrients and the oxygen that it's going to need, because that's what these red blood cells are for. They're full of that protein hemoglobin, and its job is to carry oxygen. In this slide, we're going to take a look at the left side versus the right side, and we're going to see the difference between what codes for a normal hemoglobin molecule found in a red blood cell versus what codes for a sickle cell hemoglobin molecule found in a red blood cell. So first, let's take a look at the primary structure. Here we see amino acids 1 through 7, and we see the same thing over here, but this one is for the sickle cell hemoglobin 1 through 7. And if we match them up, 1 to 1, same amino acid, 2 to 2, same, 3 to 3. And it's not until we get to the 6th position where you see a valine in the 6th position for the sickle cell hemoglobin. If you look over here, we see GLU, and obviously it's not a match. So what does that do? Well, once you start folding your beta pleated sheets and your alpha helixes, what we're going to wind up getting is a malformed beta subunit. Look at this one here for a normal hemoglobin. Then look at this one here for a sickle cell hemoglobin. The shapes are not the same. And what that's going to do at the next step, once it's folded upon itself and it's a certain shape, and we now have to put the units together to form the quaternary structure for hemoglobin, the beta units must be right. Here we have 2 beta units that are attached to it. Here in the quaternary structure of the sickle cell, we still have the 2 alphas connected to 2 betas, but again, the betas are malformed, so they're not right. And then when we get to functionality of these 2 molecules, these molecules here do not associate with one another. Each carries oxygen. But then if you look at this one here in the sickle cell, molecules interact with one another and crystallize into a fiber. Capacity to carry oxygen is greatly reduced. So for some reason, by folding these the wrong way, they're now going to fit together in this strange fiber-like form, and they're not able to carry oxygen real well. So once the entire red blood cell is built, this is what you get when it's made of millions of these molecules, and this is what you get when it's made of millions of these fiber strands. And again, this is normal shape, this is abnormal shape like a sickle, and that's not going to carry oxygen very well. So all of this boils down to one amino acid change in the primary structure. It's not just single amino acid substitutions that change protein structure. We can have some other things that are more environmental that can change the protein structure, and usually those environmental things are not helping the protein. They're usually hurting it in some way. So alterations in pH, how acidic or basic the solution is, salt concentration, whether it's high or low, temperature, is the temp too high for this protein? And lastly, environmental factors, which could be something new that this protein, this polypeptide, will now chemically react with. It's not really good for the protein. Most of these things, when they're out of the narrow range of limits they should be in, is going to cause that protein to unravel. And remember, we said structure fits function. So if it unravels, it's not going to do the job in which it's supposed to do. We call that denaturing. So when there is values that are way out of whack in whatever normal ranges, from whatever normal ranges they should be in, the protein denatures, and that makes it biologically inactive. It won't work right. So here is a normal protein. We see all the alpha helixes and beta pleated sheets are right where they're supposed to be. We hit them with one of these things here, some level that it shouldn't be at, and the thing is going to wind up denaturing. So if it denatures, it's going to unwind. It's going to be this loose polypeptide chain that really doesn't do a lot of anything good for the organism. However, if conditions do return fast enough, all might not be lost. It might refold back into its normal state, which we call renaturing. And once again, it can be and work as a normal protein. So whether a protein has denatured or whether a protein has just been built, it has to be folded properly. And sometimes, just by chance, it's not going to fold in the right configuration. It needs a little bit of help. So in that case, we call in the proteins called the chaperonins, which are going to chaperone that polypeptide and fold it into the correct configuration. So what a chaperonin looks like here is we see this hollow cylinder made of protein, has a little cap on it. Your polypeptide is going to float on in to the center core in this hollow portion. The cap is going to be put on top, is going to chemically migrate to the top of the structure. Inside of the structure, we now see that loose polypeptide has been folded correctly. The cap's going to come off. Your corrective folded protein is now going to float to where it needs to be. And that's what chaperonins do. They're going to help out fold a polypeptide correctly. Okay, you're almost there. We got one more biomolecule to go, and that happens to be nucleic acids. Now, earlier in our lecture, we asked the question of what predicted the primary sequence, the primary structure of a polypeptide. And we kind of left it open-ended, but now we get to the answer. If you look at the first bulleted point, it says the amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. So that primary structure of amino acids that were linked together by peptide bonds was dictated by a certain sequence of nucleic acids, which collectively we're going to call a gene. And genes, as you can probably remember, are made of DNA, one of the types of nucleic acid. So if we check out the little picture down here, here we have a stretch of what we can see as a chromosome here. Unwound is the typical DNA that we're used to seeing, the twisted ladder structure, and we see that a certain segment here is coded as a gene, something that's inherited from parent to offspring. So genes are going to contain the instructions for building those polypeptides. Aside from the storing of genetic information, there are some other roles of nucleic acids as well. For example, DNA will provide directions for its own replication. So every cell is going to need to make an additional copy of its DNA, because if that cell is to split, each time it split, it would only have half the genetic content it had in the previous cell. So therefore, you must double the amount of nucleic acid within the nucleus of a cell, and DNA will provide the directions for its own replication. Second, DNA will direct synthesis of RNA, a different type of nucleic acid, and through it control protein synthesis, as we said in the last slide. So what we're talking about here is a pathway that starts at DNA, a nucleic acid. It's going to then go into RNA, and then is going to be translated at a later time into a working protein. Now, this sequence of DNA to RNA to protein, this is what we refer to in biology as the central dogma of biology. It is a particular route of information from the nucleus of the cell to traveling outside that cell to be then translated into a protein and then go to work within the organism that carries it. We also see another role of nucleic acid as an energy transfer molecule. So we're actually going to use it as kind of like the energy currency of a cell as well. We'll get to that in a little bit. And then lastly, we use nucleic acid sometimes as intracellular signal molecules. So when cells have to communicate with other cells, it can be done through a type of nucleic acid. Focusing on the idea of the central dogma of biology, let's go ahead and let's follow this diagram. What we have here, this is the interior of a cell, and as you remember from your last biology class, remember every cell has a nucleus, and outside that nucleus is the jelly-like fluid that's called the cytoplasm. But let's focus on the interior of the nucleus first, and here we see DNA. Now, the first step in the central dogma of DNA is going from DNA to a copy of that DNA called a messenger RNA. So this is going to unwind and be copied into a little strand that you can see here that happens to be single-stranded. We know that DNA is double-stranded, and the helical shape kind of crosses back and forth. But RNA is single-stranded. This would be like me taking a book to a copier, popping it open to page 237, slapping it down on the copier glass, and hitting copy once. And what I get is a copy of the page. So maybe this segment here is going from here to here, and that's all I wanted. So that's what that little red strip is, one copy of one of these strands of DNA. Now, after the mRNA has been transcribed from the DNA, it's going to worm itself out of the nucleus by way of a nuclear pore, and then it's going to find a little two-part organelle within the cytoplasm called a ribosome. There, that mRNA is going to thread through the two parts, and what it's doing then is building a polypeptide. As you can see, here we have free amino acids, which we called monomers earlier. And as those monomers come into this two-piece structure, this ribosome, what we have being built is a polypeptide. And these are all linked together through those peptide bonds that we talked about earlier. So from the interior of the nucleus all the way out here to formation of a polypeptide, that is going to embody the central dogma of biology, DNA to RNA to protein. Let's focus in now on the actual molecular structure of these nucleic acids. These come in polymer form, just like we saw with our carbohydrates and our proteins. And if you notice in both of those, when we turn into a polymer, we use the word poly in front. Same thing's going to happen with nucleic acids. The only biomolecule that doesn't polymerize is going to be the lipids. So nucleic acids then will be polymers of polynucleotides, and each polynucleotide is made of the monomer, the little tiny piece that builds it up, called just a nucleotide. Now, if we look over here on the right, you can clearly see we have what is a long strand composed of many parts, but we're just going to focus in on this part from here to here. This is going to be your nucleotide. And each nucleotide is going to be made of three things. One of the things is a N-containing base. Think back to your chemistry. What does N stand for? Of course, it's nitrogen. So a nitrogenous base is what we'll call it. And this little hexagon will represent that nitrogenous base. Second, a sugar. And down here you can see this sugar, which is called a pentose because it's made of 5 carbons, is in the center. And lastly, there's going to be a phosphate group that you can clearly see here, a phosphate atom with 4 oxygen atoms around it. So it's that unit that will be repeated again and again and again that will make one of these polynucleotides. So here we have 1, 2, 3, 4 different pieces. Now, sometimes we can refer to just the sugar and the nitrogenous base, and if that happens, we're going to call it a nucleoside. So without the phosphate, just the sugar and the nitrogenous base, we're going to call a nucleoside. Add the phosphate to it, it becomes a nucleotide at that point. 5 nitrogenous bases exist. Let's take a look at them and their names. So what is the commonality between these 3 structures on the top? Well, it's the fact that each one is a single ring structure. This first one, as you can see, is called cytosine. The second one is called thymine. And the third one is called uracil. Down on the bottom, these 2 down here are a little bit different because they're made of double ring structures. And the 2 nitrogenous bases that qualify for that are going to be adenine and guanine. The 5 nitrogenous bases can be broken into 2 families. The first family is called the pyrimidine family, and it's made of cytosine, thymine, and uracil. And again, we said they have a single ring structure. The second family is called the purine group, and they have the double ring structure. So if you spot a double ring versus a single ring, you can always rule out certain possibilities for whether they qualify for that group or not. Couple other differences we see when we talk about nucleotides is sugar differences when we compare DNA to RNA. In DNA, your sugar is going to be called deoxyribose, but in RNA, the sugar is just ribose. So that's one of the key differences between DNA and RNA, a commonly asked question. Also, the nucleotide, if we put it all together, we could say that a phosphate group plus a nucleoside, remember it's the nitrogen containing base and the sugar, will equal out to a nucleotide. Take a look at these 2 sugars. They look really similar. However, they're entirely different, and they are going to be parts of different types of nucleic acid molecules. Can you see the difference? Well, look at their names first of all. Of course, they have different names. This one here is called deoxyribose, and this one here is called ribose. If we look at their structures, most of the structure looks almost identical, except in one position, and that position happens to be carbon number 2. And this one here, notice the difference between that one there on the right. There's an oxygen present in ribose that's not there in deoxyribose. So deoxy, you can think of it as like minus an oxygen. So this one here is going to be present in your DNA. However, this one here is going to be present in your RNA. So how do you actually make a polynucleotide? Well, let's take a closer look at it. Adjacent nucleotides, that is side-by-side nucleotides, they're going to be joined by covalent bonds, and they're going to be covalent bonds between 2 things. First, the hydroxyl group on the 3' carbon of a nucleotide. All right, let's stop right there. So here is our sugar, and one of the carbons of the sugar is called the 3' carbon, and it sits right here. That is going to form a covalent bond with the phosphate that is connected to the 5' sugar, 5' carbon on the next sugar in the next nucleotide. Okay, what does that mean? Well, here's the next nucleotide down. Remember, phosphate sugar base. Here is the 5' carbon, and here is the phosphate group that's attached to it. So the location it's talking about is right here. And the bond that exists between them is this little black stick right there. That's the covalent bond. And that will keep repeating itself as new nucleotides are going to be attached into this chain. And this has its own name as well. It's called a phosphodiester bond. Okay, so that's going to connect one nucleotide to the next. Review: in carbohydrates, it was called a glycosidic linkage. In lipids, it was called an ester linkage. In polypeptides, it was called a peptide bond. And now in nucleic acids, we have a phosphodiester bond that links them together. These links will create a backbone of sugar phosphate with units with nitrogenous bases as appendages. So what we have then is we have this fairly straight, however a little bit angled. We go right, left, right, left, right, left. But that's going to form the edge of one of these nucleotides when we talk about DNA or RNA. And we call that edge the sugar phosphate backbone. You've probably heard of that phrase before. Now, what hangs off into the center portion, if we're talking about DNA, are these nitrogenous bases. They're almost like the rungs of a ladder. So the DNA double helix then has two polynucleotides spiraling around an imaginary axis, forming what we said was a twisted ladder. And as you can see, here's an example of a twisted ladder. In the DNA double helix, the two backbones run in opposite directions to each other. And that directionality is called the 5' to 3' direction, and we'll see that in a second. When we have two of the sides of a double helix running in opposite directions, we're going to call that anti-parallel. So one goes top down, the other one goes bottom up. Another thing that we can say is that DNA molecule, we mentioned what a gene was a little bit earlier. One DNA molecule is going to have many genes that occur down the length of that polynucleotide. Now, a couple of extra details we'll throw in here. As you can see at the last bullet, the nitrogenous bases, those parts in the middle, in DNA are going to pair up and form hydrogen bonds between the two sides if we're talking about DNA. And what we're going to see first is the the nitrogenous base thymine, which we represent with just simply a T, is always going to bond with adenine, which is going to be the A. And the other combination is going to be when we have cytosine, which we can use the letter C for, bond with guanine, which is going to be G. And again, this is for DNA double helix. It's who connects with whom as we go down a double strand of polynucleotides. When we have A is always bonding with T's and C is always bonding with G's, it's called the law of complementary base pairing. This slide here shows you the anti-parallel nature of the two sides of a DNA molecule. Here we have our phosphate sugar. So this is our sugar phosphate backbone. In the center, these are our nitrogenous bases that are sticking out. And we said a 5' to 3' direction is the way that it's arranged, which the other side will be the opposite. So here you have your 5' and here's your 3' end. How do these look different? Well, if you look at the one on the right, notice that it's flipped upside down. So it's not as if it's perfectly arranged in a line where phosphate's directly across from phosphate and sugar are directly across from sugar. Instead, because this strand here is upside down, there's a little bit of difference in the positioning of those different parts of the nucleotide. So 5' to 3' in one direction goes 3'5 to 5' in the other direction, and that again is called the anti-parallel nature of the double helix. This picture here is interesting as well because not only does it show the 5' to 3' nature, but it's also showing you a DNA that is replicating itself at the current time. So let's take a look just for a second at the 5' end, which you can see right here. If we go ahead and we follow it down, and what I'm following right now is the sugar phosphate backbone, all the nitrogenous bases are represented by single letters coming off of them. And notice that it's going to keep coming down here, and it's going to wind up at a 3' end. I started with the 5' and I ended with a 3'. Now, the opposite strand, notice that here I have a 5' end, and this one again goes the other direction, and since it's being built, it's going to stop right here at its 3' end. Again, we can look at the other strand over here. This is the 3' end, the anti-parallel end, And if we track this one all the way down here, we should reach a 5' end down here, and the opposite complement to that would be a 3' end. This one is also being built at the current time, and it will end in a 5' end. So even when it's splitting down the center, it will still follow that anti-parallel nature. So that's DNA, but what about some other nucleic acids? Well, we mentioned one earlier called RNA, which is called ribonucleic acid, and it's composed of a pentose sugar, which we said ribose, not deoxyribose, still has a phosphate group, and still has nitrogenous bases. So if you look at the molecule over here on the right, here's the sugar phosphate backbone, there's the nitrogenous bases sticking off, but if we take a closer look upon inspection, you're going to notice that there's U's in here. We're not going to have U's in DNA, but we will have them in RNA, and that represents uracil, and it takes the place of thymine in RNA. We also notice that it's single-stranded. There's not an equivalent polynucleotide on the other side, making it a double helix. This is a single helix. And we mentioned already that the bases for RNA are adenine, uracil, guanine, and cytosine. Again, uracil replaces thymine. RNA molecules will often serve as a temporary copy of a certain segment of DNA that maybe it's going to be translated into a polypeptide a little bit later. Some RNA molecules are what we call regulatory, acting as enzymes, facilitating some chemical reaction to happen, or possibly silencing gene expression. So sometimes genes will be expressed fully in an organism, and often they'll be quieted down by certain RNA molecules that will provide interference for that expression. Another role that nucleotides have in the body is related to energy. We call ATP, adenosine triphosphate, the energy currency of the body. And if we take a look at its chemical structure, down below here we can see a nitrogenous base. Here's adenine, here's ribose, the sugar present in RNA. But then over here, it's not one phosphate, but we have 3 in a row. And what this works like it's kind of like a little battery, and what you get to spend from an ATP molecule are these phosphate groups right there. As you spend them, you then create energy inside of a cell with a chemical reaction. So very simply, you can think of ATP kind of like a rechargeable battery, whereas if you spend the phosphates from that chain on the end of the molecule, you'll go from a charged state to an uncharged state. Well, how do you recharge your ATP molecules? Of course, we eat. And as we ingest food and we break it down into little pieces, the glucose from our food will eventually fuel the creation of ATP. Fats can also fuel it, and so can protein. Here is the molecule a little bit more precise in terms of the chemistry that makes it up. Here are your 3 phosphate groups in ATP. Here is one that has lost one of these phosphate groups. This one has gone away, and this one is called ADP, adenosine diphosphate. So this would be like a battery that has lost part of its charge, and to recharge it, you're going to have to create more ATP, and another phosphate group will then attach to that ADP. Okay, so let's put it all together. So not only are nucleic acids the storehouse of genetic information, but they also provide the molecule for cell energy production. We mentioned the fact that there's 3 phosphates that are attached to the molecule called ATP, and as you break or cleave those phosphates from that molecule, that's going to release energy. And that's going to do the body's work, basically. So what we see then is we see ATP, and here is kind of a different look to it, but we have the adenosine and we have the 3 phosphate groups. We can go ahead and we can cut one of those off, and by cutting it off and making it ADP, adenosine diphosphate, we now have created energy. So ATP is a remarkable molecule in the fact that it can release energy for use in anabolic reactions, building other things while you're breaking that down, because we can always replenish the ATP with energy from food. Now, we even have an alternate system for when ATP gets depleted too quickly. We can go to the alternate system to fuel whatever needs we have, and that's done through a chemical called creatine phosphate. Or we can even break down one more phosphate group off of ADP, and then it becomes AMP, which is called adenosine monophosphate. That's going to conclude our screencast on the biomolecules. In the first part, we took a look at anabolic versus catabolic reactions, how they happen. We focused in on dehydration synthesis versus hydrolysis. Then we took a look at carbohydrates, and we also took a look at lipids. In the second part, we started with proteins. We looked at peptide bonds. We talked about how they were made and how they were broken down. And we looked at the 4 different levels of protein structure. In the last part we just finished, we talked about nucleic acids. We talked about the fact they can code for genetic information. They can also fuel the body through ATP molecules. So I hope you learned a lot about biomolecules today. You'll be putting this to use in later chapters to come and later lectures, so it's good that you have this right at the beginning so you can form a base for adding new information onto it a little bit later. So make sure you do your reading to glue this all together, and I'll talk to you next time. See ya.